is red, so on and so forth. And so the question is, what gives an organic molecule color? So to have color, the molecule must have a chromophore. And that's a functional group that's going to absorb light so that, so that we can perceive what that light is. So in regards to this discussion, the chromophore is a conjugated system. So in terms of that, the minimum is sort of two double bonds next to each other, and then we're going to grow that out to increase conjugation. So one thing I can say, the more unsaturation, so the more double bonds, the more intense the color. So the bottom circle, anyone take like Art 101? So your gen ed requirements. So I took some art classes in college, and they teach you about color theory. Have you heard about complementary colors? Like with fashion, you want to pair certain colors so it doesn't look tacky. So in the color wheel, complementary colors are basically 180 degrees from another. And so like if you think about a lot of sports teams, their uniforms are complementary colors. So what I mean by that, if we see orange right here, its complementary color is blue. So we would sort of pair those together. And then you can use these other qualitative words like warm and soft and that sort of thing. So color is very important in our life, whether you realize it or not. And it's as humans, we've developed these standardization scales. So back in the old days, I don't know even know if there's one up here, but my iPad's connected by an HDMI cable. But before, like if you have an old Nintendo, there were like different cords for the colors, you know what I mean? So red, green, blue, that's one scale. Anyone have a Hewlett-Packard inkjet printer at home? So cyan, magenta, yellow, black, that's another scale. So you should all be like angry right now because like if you have one of those, if this ink pod runs out, then it won't print your black text just because that runs out. And it's like you got to replace that to even print anything. So it's a grift. Another way of describing color is hue, saturation, and brightness. And then probably more of like the numeric scale, the hex codes. And if you look on here, I use Apple. So this is the Apple color palette with crayons. These are the sliders. You can move those or you can enter numbers and it'll spit out the hex code. So that's kind of interesting to think about, that a number can represent a color, and you can, whether you know like different, how many, did you have the 8 crayon box or the 64? How many color names do you know? And then what marketing people use is Pantone. So if you go to like Lowe's and you want to paint your bedroom, you get those cards. That's the Pantone scale. And so there's a lot of different colors available to us. So I think I already answered this one. A compound that appears to be orange will absorb what color light? So this is another weird sort of thing to think about in terms of what you perceive as color. So a compound that appears orange, what ends up happening is the substance absorbs blue light. And so the complementary color is what you see. So this would be A. So, that's a crash course in color theory. So how does this relate to organic chemistry and organic molecules? So I like to sprinkle in these slides about Nobel Prizes because I think they're really milestones in human intellectual progress. So in 1937, Hayworth was awarded the Nobel Prize in Chemistry for his investigation into carbohydrates. We'll see his name pop up again in Module 5 when we talk about biomolecules. In vitamin C. Carrar for his investigations on carotenoids, flavins, vitamins A and B2. And then in 1938, Kuhn won for his work on carotenoids and vitamins. So this was an era where people wanted to know the structure and function of these molecules. People were really thinking about, okay, like why is a carrot orange? Why is a tomato red? Like what gives it those properties? So we'll look at a couple classes of these molecules that have chromophores in them and what gives rise to their color. So carotenoids are polyunsaturated hydrocarbons, and so I'm going to use this word tetraterpenoid. At the end of the lecture, we'll talk about what terpenes are. And they're typically yellow, orange, and red in color. So of the carotenoids, there's carotenes. So those are just pure CH compounds, carbon and hydrogen only. And then xanthophylls, which contain oxygen. I took biology in high school. I didn't take it in college. I didn't take it in graduate school. So some of this stuff, if you've taken biology, you should start making some connections. If you're thinking about plants and why plants have colors, you might have heard some of these terms already. So carotenoids absorb in wavelengths ranging from 380 to 565. And so because they absorb there, they appear yellow, orange, and red in the complementary regions 565 to 740 or just 800 to make it easy. So we'll talk about some of these things. We'll identify the structural features, and then we're going to quantify it a little bit because we are talking about spectroscopy. And again, to refresh your memory, the word spectroscopy means the interaction of light with matter. If you are taking a pre-professional exam like the MCAT, spectroscopy is going to be very important to you. Medical imaging is spectroscopy. So the molecule shown is called lycopene. It's a photosynthetic pigment. So there's a word pigment. When we think of that, we think of substance that's added to another thing to give it color. So it's a polyene, so that just means many alkenes, responsible for giving the red color to fruits, such as watermelon, grapefruit, and papayas. So I have a question for you. Do you notice any particular repeating pattern? What do you think? So let me first highlight the conjugated part. So remember, conjugated bonds are only separated by a sigma bond. So even though it's starting with a pi bond there, that doesn't count because you have sp2 and sp2. So the conjugation would start here and then move along this backbone. So that's the conjugated part. And notice it's only carbon and hydrogen. And so if you're visually clued in, what the repeating pattern is, in the methyl groups are really the milestone that sort of give it away. So the repeating pattern is this. So that 5-carbon pattern, the repeating unit is called isoprene. So really this is a polymer of isoprene. And if you go on, to take other cellular and molecular biology courses, or say like a medicinal chemistry course or something like that, basically what nature is doing is taking an isoprene derivative and then through a series of enzymatic reactions is polymerizing it. And so that's how you can get a lot of carbon. So what is there? There's 40 carbons built up pretty quick. So the break in the pattern is right here. So notice on one side, sort of all the methyl groups are on top. On the other side, they're on the bottom. So at that point, something happens in that enzymatic cascade that's different. We'll talk a little bit about enzymes in Module 5, and enzymes are just big organic molecules. So lycopene is transformed into beta-carotene, so this is a new molecule, by an enzyme called lycopene cyclase. And just to give you an introduction on that nomenclature, typically things that end in ASE are enzymes. And then the prefix of the word is the action of the enzyme. So cyclase, that means it's taking something linear and cyclizing it. How that happens can be a little bit more complex, but ultimately that's what it's doing. So take a snapshot of that in your brain. Boom, that becomes this through an enzymatic reaction. And so now this is beta-carotene. And so it's a polyene responsible for giving the orange color to vegetables such as carrots, sweet potatoes, and cantaloupes. And all we've done, again, was remember that alkene at either end that was not conjugated? It's cyclized now. And so that you can see that the backbone is fully conjugated. So now you don't have any other double bonds or any breaks between them. So at this point, we're really departing from my own knowledge or interest in things. When I say carotenes transmit energy, they absorb to chlorophyll. So these things exist like in plant leaves and whatnot. And so the leaves get energy from sunlight. And so that energy is transformed through the electron transport chain. And so then you're into the biological processes at that point. But again, the reason that these exist in plants and whatnot is so they can absorb that sunlight to give them energy. So what about things that are not red or orange or yellow? So blueberries, black raspberries, eggplant, grapes, red cabbages. These have much different structures in them, so that just visually should look a lot different to you. It's no longer like a linear chain with conjugation, but now you have these rings. But when you look at the double bonds in those rings, they're all conjugated. And so this has like an oxygen plus, and then there's some counter ion that's balancing that out. So the darker coloration these are called anthocyanins. And the core here this is called flavillum cation. And so this is an interesting word here, halochromism. So what's the name of the flower that like changes color depending on like the soil pH? Hydrangea. So it can be like what, pink or what?
Blue or purple or white.
Blue, purple, or white. So based on that structure, the pH of the soil will change the nature of that compound and then the color of that flower. And so that's, like if you think of it in the scientific sense, it's a sensor of pH. So it gives you, is this good soil to grow this in or not? But again, color is very important in our lives, and I'll try to make that abundantly clear to you as we move on. So again, from the food, the natural food, fruit and vegetables we eat, we see that color. I'm sure a lot of you have a meal plan. You eat it fresh. Do you eat your veggies and fruits every day? Are you getting your nutrition in terms of the food pyramid? You're eating other stuff. And that stuff typically has artificial color added to it, so these dyes. These little bottles you see, we used to buy them for Easter to like color eggs, you know what I mean? So again, the way this works is that, in the Roy G. Biv here, so that the dye is absorbing the complementary color, and then what is transmitted is what you see. So because we see blue, it's essentially, absorbing the complementary color orange. And for the red, it's absorbing in the Biv region, so we see the Roy in there. So try to have a basic understanding of the wavelengths that are being absorbed versus what you're seeing, which is what you're perceiving as color is the light that's being transmitted. So I mentioned artificial dyes because a lot of the things we eat, like especially candy, like licorice, like red licorice. Some people like it or hate it. It's red because there's a dye added to it. And so these are common food dyes. They're normally referred by like the color and then some number. So like yellow dye number 5. Anyone like M&Ms? So M&Ms have that shell because there's a food dye added to the candy part, and then there's the chocolate center. So, there are some alternative names, the color, and then the molecular formulas are pretty big, right? So here's one specific red dye number 3, and I mention this because the US FDA recently banned this substance. So the point I'm trying to make is that color is added. Why do you think color is added to food?
Make it more appealing.
To make it more appealing to you, right? So generally, if there's more color on the plate, that makes you hungry, so you eat, then you're not unhappy because your belly's happy. But in reality, the color doesn't need to be there, right? It serves no nutritional value. It's not giving your cells any energy. And so the reason that red dye number 3, so it gives it a bright cherry red color to things, was banned is that there's studies that showed it can cause cancer in rats. And so the rat model is kind of used. You can't do this stuff on humans, right? So it's tested on rats, and then you project out into other species. So it was banned as a food additive, a couple weeks ago of this year. And then the manufacturers that use it have 2 or 3 years to comply based on what industry they are, whether it's food or drug manufacturing. An interesting thing, when my son got sick and I had to take him to the pediatrician, they gave him this cough syrup, which tastes bad to begin with. So they added a color dye to it. It was red, and it had like a cherry flavor, but he absolutely did not like that either, to the point of like gagging on it. So it's like the stuff comes as originally like a white suspension. They add a red color to it, and it doesn't really change it, right? So the color is just there to engage you. And so I've opened up a bonus in Canvas that'll be due next Monday. Take a picture of something you've bought and just upload it. And there's questions for that prompt. You just got to upload the picture. Why did you buy that thing? Like, what attracted you to that product? So an interesting experiment to think about your own susceptibility to psychology. If you're ever in Walmart, walk down the laundry detergent aisle and look at the boxes and the containers. How many different colors are there? There's so many, right? And so, if there's 20 different manufacturers making this stuff, how are they going to get your attention? The shape of the container, the color, the font that's used, the size of the font, all of that stuff. And the color is what is grabbing your attention. And at some point, we're all sort of weak-minded. we have to buy shit, right? So and then it really gets into your own psychology. What do you prefer the most? So as I was looking at my can of Red Bull, I drink 2 or 3 of these a day. I got to get through it, you know what I mean? Otherwise, I'm going to fall asleep in my chair. You know what it says on the ingredients? It just says colors. That's all it says on there. So
It doesn't tell you what colors.
it doesn't say what colors. So if you're looking at something you bought and it's food, look at the ingredients and see if it lists like what is in there in terms of dyes or whatever. If it's like a piece of clothing, like why did you buy it? Probably because it's your favorite color. If you're wearing like a white shirt or something, it's not dyed. That material is whatever it's made of. And we'll talk about those things in Module 5 when we talk about like polymers of glucose. So cotton fiber is a polymer of glucose. It's a really long chain that forms a thread. There's a lot of those threads, and they make clothing. And so cotton is white, so it gets color through these dyes. And if your clothing is not 100% cotton, then it's like polyester, which is a polymer of some probably amide or ester. But it's still getting a synthetic dye put into it for color. And that, again, it impacts our lives quite a bit in terms of our preferences, what we buy, what we eat, so on and so forth. Any questions? So a couple more examples. This is chlorophyll. And the conjugation part is in the large ring. So when I say that, you have these smaller nitrogen containing rings. They're called heterocycles. But then you have since they're all bonded to one another, you have a large ring. And so that's the conjugated part. And so that forms something called a porphyrin. And then since you have the nitrogens in the middle, we know they have lone pairs, and then you can get these metals that sit in there. And so that's what I was talking about, converting sunlight energy into the electron transport chain. So we'll talk about Emil Fischer in Module 5 in terms of glucose, but another Fischer, Hans Fischer, won the 1930 Nobel Prize in chemistry for researches into the constitution of Haemin and chlorophyll and the synthesis of Haemin. So I don't have it on there, but so this is in plants. Do you know what's in humans? Huh? Not a sugar. Heme. So yeah, within hemoglobin is a molecule that looks like this called heme. And then it doesn't have magnesium in the middle. Do you know what it has? Iron. So iron is the metal in heme that's in your red blood cells. So everyone take a deep breath. So you're sucking in oxygen. That oxygen goes to the iron. And then from there, it's transported throughout the body. And again, in Module 5, when you exhale, do you know what you're exhaling? Carbon dioxide. So if you're breathing in oxygen, where does the carbon of carbon dioxide come from? Glucose. So the food you eat gives you glucose. The glucose is metabolized in the presence of oxygen to give CO2.
So what happens if you stop eating?
You will die.
Obviously, but there's no, never mind.
Again, I did not go into biology too much, but probably what will happen is, your blood is a certain pH, right? So I would assume that if you're not getting glucose, that pH is going to change to a point where your cells basically, through some processes, start dying. So a little bit of math, not much, to quantify. Looking at an organic molecule, can we predict where it's going to sit on the visible spectrum? How can we calculate the wavelength of light we'll see? And so this is done through UV vis spectroscopy. So this is electronic spectroscopy. And really what that means is how electrons are moving from one orbital to the next. So you get energy coming in, electrons are promoted into different orbitals. So we know what those orbitals are. So the change in energy, delta E, promotes an electron to the highest occupied molecular orbital. The way to think about that highest occupied molecular orbital is like when we're talking about reactions, and we say a nucleophile is supplying electrons, those are the electrons available for bonding. So that's the highest occupied molecular orbital. So promotion of an electron from the highest occupied molecular orbital into an orbital that's unoccupied. So lowest unoccupied molecular orbital. So the electron hops up, it then goes back down, and as it goes back down, it's releasing energy in terms of light. So there's some equations. You'll probably apply this more in physics than you will here, but I'm showing it to you. There it is. in terms of the instrument, that's what it looks like. Basically what happens, there's this part right here. This part right here I'm highlighting is basically that. You get this thing called a cuvette. You make a solution and you just stick it in there. And the way the instrument works is you have a reference beam. So it's when light can go all the way through, it's not hitting anything. And then you put your sample in, and the light is interacting with that sample, and it's going to come out as a different intensity. And so it's A equals the log of the reference beam divided by the sample beam. And this is called Beer's law. So epsilon is something called molar absorptivity. And that's really the key thing for this equation. That number differs for every compound. B is the length of the cuvette, which is usually 1 centimeter. And then C is the concentration. And so again, A means absorbance. So how much light is being absorbed by the compound relative to the reference beam? So again, as what we're talking about, there's the visible region. Again, over here is the UV part. Over here is the IR part. And this is what we see. You know, we can't see UV light. We can't see IR light. We just see the visible part. And what we're aiming to understand, where along this part of the spectrum will a compound transmit light based on the extent of conjugation. So we're going to think about this in terms of going from the left to the right. So at the left here, we see ethene. And let me just say here, shorter wavelength, higher energy. So ethene is a gas. It has 1 double bond. So what's happening is that we know that in the pi bond there's 2 electrons. The pi bond contains the highest occupied molecular orbital, so 2 electrons. When energy hits that, one of them gets promoted to the lowest unoccupied molecular orbital. So the difference between the highest occupied and the lowest unoccupied is an energy difference. See that number here? We call that pi to pi star, and I have another slide that explains that. That energy is 700 kilojoules per mole. As you move from left to right, what does that number do as the compound adds more double bonds? The energy decreases. So what that's telling you is, as you add more double bonds, it's easier for the energy coming in to promote an electron. And as you do that, like, see this number 171 nanometers? If we extend this scale, that would be like here in the ultraviolet region, you couldn't see it. But as you're adding more and more, you're pushing it into this region, and now you can start to see it once you break that threshold. So really we see Yeah, I guess what I want to point out here is pi star decreases with increasing conjugation. So if you can, again, picture lycopene in your mind, it had, all those double bonds in it. It's sitting over here. So that's telling you, or you can infer, there's a lot of conjugation to it. So the energy gap, here we're starting with just ethene. I didn't put the structures on here, but this is just ethene. This is 1,3-butadiene. This is the triene, and this is the tetraene as we're adding more double bonds. Because ethene, when you make that pi bond, you have a p orbital and a p orbital. So that's 2 orbitals, right? You get the bonding, that's the highest occupied. So the electrons are occupying that. But since you start with those 2 atomic orbitals, you have to produce 2 molecular orbitals. So this one is called pi star. So that's an anti-bonding orbital. And this is the energy gap. So it's big. When you add the second double bond, the energy gap starts decreasing. And so it gets to the point that, as I said, the gap is so small you don't need a whole lot of energy to do that promotion. So I assume you all have flat screen TVs. You obviously have phones and tablets and computers. So have you heard of the term OLED? What's the O stand for? Organic light emitting diode. So I'm sure there's some polymer students in here. A lot of polymer research is making polymers that are organic light emitting diodes. So along that conjugated backbone, you're sticking in substituents with the goal of controlling this gap to a very specific energy so that the transmitted light has a certain wavelength. In the lower energy level, they're all spin paired. When this gets promoted, it's still retaining that spin. So if I call that spin down, it ends up as spin down. And this is essentially what happens when that energy is added. You get that promotion of an electron from the lower energy to the higher energy orbital, and notice that the spin is conserved. If you're, I don't think anyone in the chemistry or biochemistry department does this research, but probably in polymer. So the spin's conserved, and then once it's up there, what they'll do is try to get this to flip. And then once that flips, it can go and do other things. And so you can control whether it's going to emit this sort of light or that sort of light. That's all I'll say about that because it's sort of beyond my expertise. Yeah.
What do you mean by flip? What flip?
See how these are paired? So on the prior slide, so it's still that spin. You can apply some other stimuli that'll flip that spin to spin up. And now that's changing the energy state. Yeah.
I just want to confirm, so like the light that we see, is that the frequency at which those electrons jump orbitals, or the energy that like caused for the electrons to jump?
I guess to sort of hedge my bets, you can sort of infer it as both. You know, it's giving you like, I don't know, there's like a student with a red sweatshirt on. You can sort of infer what that energy gap is that's leading to that red color. So what we're going to do now is look at something called the Woodward-Feser rules. So again, there's Woodward's name. Again, Woodward was a synthetic organic chemist at Harvard. Feser was his colleague. And an interesting factoid, Feser was the guy that invented napalm. So you know what napalm is? So it's jellied gasoline. If you take gasoline and add like dish soap to it, it sort of gives it like a jelly consistency. And then you can stick it in a flamethrower and like burn down forests and other stuff. Again, you know, at that point, sort of in history, scientists were recruited for war efforts and applied their research to that. But based on Woodward's interest in synthesizing natural molecules, you know, he started to notice patterns in the molecules he was trying to synthesize. And then he worked with Feser to develop this set of empirical rules. Empirical just means based on what they're observing. It's not coming from theory at all. And really, what it's used for is to estimate lambda max. What is the wavelength at which we should see color from this conjugated system? And to show you the empirical part of it, you know, starting from 1,3-butadiene, they figured out that that's the minimum requirement to start conjugation. That gives a base value of 217 nanometers. And then when you start adding double bonds onto that, you get sort of a consistent value for each double bond you add. And so that's the empirical part. So you add one more, you get 258. You add another, you get 290. And then you can start decorating that chain with other things and how will they affect what sort of standard value should be added to 217. So the Woodward-Feser rules are a way to estimate the lambda max. Where is the light going to be most absorbed in UV vis spectroscopy? And the starting point, so I'll do it in red here, is again 217. For every double bond that you add, you tack on another 30 nanometers. And then if you substitute hydrogen for something else, that's called an oxochrome. And each of those would add 5 nanometers. So 217 plus 5, that gets us what? 222. So that's basically what we're doing. So we added that methyl group as an oxochrome. The base was 217. It contributed 5, so we get 222. So here's our starting base structure, that dying. If the dying happens to be outside of a ring, that's going to add +5. So you're sort of looking for that substructure in whatever molecule you're considering. If the dying is locked together, so that would be like the S-cis, that's called homoannular, and you add 39 nanometers to the 217. If you're in the S-trans, this is called heteroannular, and you're starting at 214. So let me just write these words down here. So S-cis, S-trans. So given this molecule, 2,3-dimethyl 1,3-cyclohexadiene, what would you estimate lambda max to be? So really it's about identifying, what are you starting with? Are there oxychromes? Then add those. So here we can see that starting diene, it's tied together, so that's homoannular. And then we have 1, 2 oxychromes. So I'll say diene equals 217, homoannular equals 39, oxychromes 2 times 5, that gets you 10. And then when you add that, you get 266 nanometers. Yep.
Is there any instance when you would subtract?
You're pointing out my deficiencies. In terms of your textbook and what I know, I only know in terms of like addition. I'm not sure if a substituent, when it's on there, if it would subtract or not. But for what you guys need to know for an exam or whatever, nothing would subtract. So using the Woodward-Fieser rules, estimate lambda max for lycopene. If each additional double bond adds plus 30 nanometers to the base value of 217, what is the estimate? This is for only the pi bond contributions. The next question regards the oxychromes. So we're starting with 217. Then for every additional double bond that's conjugated, that's adding plus 30. So in the conjugated system, how many total double bonds are there? Huh?
9.
So the answer is 9. How did we get there? Well, I mean, the final answer is 9. The total is 11. So this is what we're starting with. So that's your 217. There's 11 total pi bonds. So we have to subtract the diene that we're starting with. So we get 30 times 9. That's 270. And we should end up with 487. So that's just the base value of the carbon chain. But remember, things that are sprouted off that are counted as oxychromes.
So why do you not count the double bonds on the ends?
So why don't we count them?
Yeah.
Anyone? They're not conjugated because they're broken up by sp3 hybridized carbons. So how many oxychromes do you see?
Six.
6 would be the probably best first approximation. So 1, 2, 3, 4, 5, 6. But it is 8 because that's 7 and 8. So that would add plus 40 to the existing 487. Question?
Can you go back to the last one for 1 second? I have a question.
The last slide,
Yeah.
this one. Good? Okay. Questions? Yeah.
Can you please go back to the other slide?
So in the last 14 minutes here, we're going to transition into another topic that's related to this, and it's called terpenes. And to preface this, the 1939 Nobel Prize in Chemistry was jointly awarded to Adolf Butenandt for his work on sex hormones and Leopold Ruzicka for his work on polymethylene, polymethylenes and higher order terpenes. So there's that word terpenes. So polyenes also go by the name terpenes, and then like the general class of these things are called lipids. So you know what a lipid is? So, you know, if you're in biological sciences, you've probably heard of the lipid bilayer. So that's the like what separates the outside of a cell from the inside. So you have like polar head groups, then long chain carbon, then a separate molecule long chain carbon in a polar head group. So that's the lipid bilayer. And so all of these things are synthesized through what is called the Ruzicka isoprene rule. And so isoprene is 2-methyl-1,3-butadiene, which is shown here. And this was, you know, really another empirical observation at this point. They started characterizing these molecules that had these common structural features, and were able to deduce, okay, where do these things come from? And so isoprene is the smallest sort of monomer in these biopolymers. So terpenes and terpenoids So terpenoid means, as we'll see, there's certain carbon counts to terpenes. a terpenoid either does not obey that count rule, or it has other atoms in it, such as oxygen. So they're produced in trees and plants and have many uses in modern civilization. Natural rubber, shown on the left, is the Z repeating unit in that polymer of isoprene. Gutta percha is the E repeating unit. And so what do I mean by repeating unit? So to see this N here, that subscript N, polymer chemists will measure out what's the average length of one of these chains. You can think of this as like a really long chain. The square bracket is just showing you what is the repeating unit of that chain. So we can highlight there's the isoprene units. And then the difference between these two is just the geometrical isomerism about the double bond. So again, Ruzicka formulated the biogenetic isoprene rule that states terpenes are polymers of isoprene bonded in a head-to-tail manner. So isoprene will bond with itself in certain constitutions, and then depending on the number of isoprene units, that's where you get these classes. So if you take two isoprenes, that's called a monoterpene. So 2 times 5, that gets you the 10 total carbon atoms. Sesqui means 1.5. So you have 3 monomers, 15 total carbons. So the trend is basically 5. So 10, 15, 20, 25, 30. And you've probably heard some of these prefixes in terms of like celebrations. So sesqui, di, sester, tri. You take that number, multiply it by, if you understand the prefix, you multiply it by 5. That gets you the total carbon count. So that's just vocabulary, nomenclature. Let's see where we see them. So terpenes are hydrocarbons or derivatives with oxygen that are isolated from plants. Since they don't have a whole lot of polar functional groups, they tend to be volatile. And so that means you can smell them. So if you look at the structures here, these are called essential plant oils. Essential just means that's the main component that's giving the plant that smell. So oil of lemon, that's called limonene. Beta-pinene, that's oil of turpentine. Like if you've ever cleaned floors, you've used pine sol. I mean, it's pretty potent, right? We're all sophisticated humans in this room, and you'd never crack open a bottle of pine sol and drink it, would you? Back in the day, people used to drink turpentine to get sort of the euphoria similar to alcohol. And then, of course, your insides would rot and you would die. So geraniol is the oil of roses, and menthol is the oil of peppermint. So anyone wear cologne, perfume? I'm a dumb guy from Wisconsin. It's cologne. So that the flavor and fragrance industry They'll use essential oils, but also they'll use like synthetic compounds as well. So, there's these stores you can go in. It's like an apothecary. It's like 500 glass jars. You want a little bit of this, a little bit of that. You put some drops in. You get your perfume how you want it to smell, or your cologne. So those are pretty big industries that utilize these molecules. And so as humans, we have deodorant, we wash. It was not always that way, right? I mean, at some point in time, your stick of Old Spice didn't exist. So eventually, people got fed up. It's like, you stink, dude, like, get away from me. And so that's what spurred a lot of this, right? So we can be in all the same room together. So this slide is a bit old. I don't know if this statement is actually true anymore, but a couple of years ago, the FDA wanted to ban menthol in cigarettes because it's an additive, and nicotine is addictive to begin with, but the menthol makes smoking more palatable, I guess, and that was one of the things that people thought it increases addiction. So in the structure of menthol, what class of terpenoid is it? Yeah, so this one's mono. So how do we know? If you count the number of carbons, there's 10. But really, the way that I do this is I'm looking for something that's going to help me figure out where the first isoprene unit is. And remember, in isoprene, it's 2-methyl-1,3-butadiene. So you're looking for sort of 2 methyl groups, and I see that right here. And then we need 5 total for one of the monomers. And so that's going to connect here. And so there's 2 of those monomers, 10 total carbons, so it's a monoterpene. And if you're really into plants and cellular and molecular biology as well, again, the plant is going to take that isoprene derivative, add another one, cyclize it, and then oxidize it. So there's these enzymatic reactions that happen to get you to that point. So zingiberine is the constituent in the oil of ginger. What type of terpene is this? So yeah, this is sesqui, it's 1.5. So there's the methyl groups, 3, 4, 5. There's the start of the next one. So there's the 3 isoprene units for a total of 15 carbon atoms. So how many isoprene monomers are in lycopene? So again, if you're looking for the methyl groups, is sort of where to start. So 1, 2, 3, 4, 5, 6, 7, 8. So a couple more slides just to finish this up. This molecule is called squalene. It's a precursor to the synthesis of cholesterol, which is a steroid. What class is squalene? I guess I heard a whisper, try. So 1, 2, 3, 4, 5, 6. So 6 means it's a terpene. So to wrap up, I want to go over, there's enzymes in your body, and I think it takes about 30 unique steps to take squalene into cholesterol. Squalene is converted into, so this acyclic thing, so this is the steroid nucleus. So there's A, B, C, and D rings. So if you take this and draw it in the chair form, that's how it looks. So again, in the 40s and 50s, people were interested in the stereochemical relationship of all these groups, these wedges and dashes, whether they end up axial or equatorial. And so you have enzymes in your body that take squalene, will put it in a conformation so that it cyclizes into that nucleus. And then after that, you have rearrangements and oxidations that occur. So there's the structure of cholesterol. That comes from squalene. And so your body produces cholesterol. It's embedded in the lipid bilayer. It's used for other things. You also get it from your diet as well. So we'll end there.